So here the continuation portion of the TSI, that is triple sugar iron agar medium. Here the glucose which is present only in 1% in this medium. It is utilized first by the fermentative organism and the entire medium becomes acidic. That means color changes to yellow within 8 to 12 hours. And the bud remain acidic even after 18 to 24 hours incubation. Bud means lowest portion incubation period because of the presence of this organic acid resulting from the fermentation of the glucose under anaerobic condition in the butt of the tube. So the butt is the lowest portion where the anaerobic fermentation takes place. Here also it may appear yellow in color. The slant portion, now it can be reversed to alkaline, that is red state because of the oxidation of the fermentation products. And sometimes if the organism present undergoes an oxidation purpose process, so the slant may appear alkaline, which means it converts to red state because of the oxidation of this fermentation products under aerobic conditions on the slant. So oxidation which takes place under aerobic condition. So the slant remains red color. This change is the result of what? Formation of carbon dioxide and water and the oxidation of the peptones present in the medium to alkaline amines, which changes the color to red. When in addition to glucose, lactose or sucrose are fermented, what happens? Large amount of fermentation products are formed on the slant. So it may neutralize the alkaline amines which appear as red color. And then the slant may appear acidic or yellow in color provided the reaction is red within 18 to 24 hours. If the slant and butt remain alkaline, what happens? The glucose has not been fermented. From that we can indicate that glucose and lactose has not been fermented. Glucose has not been fermented. That's why both slant and butt appear red in color. That means alkaline. Organisms showing this reaction are defined as non-fermenters. Non-fermenters are those which doesn't ferment glucose. So the butt and slant remain red color, no color change. They are called non-fermenters and they derive the nutrients from the peptones which are present in the medium. Next is the formation of this carbon dioxide and hydrogen gas by some organisms. It is indicated by the presence of bubbles or cracks in the agar. Sometimes the agar can be formed the crack and bubbles. That is indication of the carbon dioxide production and hydrogen gas. Or the separation of the agar from the sides or from the bottom of the tubes. So it may indicate the gas production, etc. The next is another indication is the production of hydrogen sulfide, which means sodium thiosulfate reduced to hydrogen sulfide. It requires an acidic environment for this hydrogen sulfide production. And the reaction with ferric ammonium citrate, it produces a blackening of the agar. But here, the ferric ammonium citrate present in the medium converts the media to blacken. As a result of the hydrogen sulfide production, the media now becomes blackened at the agar butt. Butt becomes black in color due to the production of hydrogen sulfide. This is the different types of reaction. A by A, which means acid slant and acid butt. This is butt portion. That is A by A. That is acid production, fermentation of glucose, lactose and sucrose. Here, A by A gas, which means acid slant, acid butt with gas production. That means bubbles. Here, A by A hydrogen sulfide. Here, acid slant and acid butt with a blackish color. That is hydrogen sulfide production. Next is acid slant and acid butt with gas and hydrogen sulfide. So air bubbles along with the hydrogen blackening of the medium also appear. Here look, acid slant, this K means alkaline butt. This is another reaction in which the organisms can be oxidative, decarboxylation. Here this is K by A. K means here this is A by K. This is K by A. K means alkaline slant, acid butt. Here K by A gas. Alkaline slant, acid butt with gas production. Here K by A hydrogen sulfide. Alkaline slant, acid butt with hydrogen sulfide production. Here K by A with gas and hydrogen sulfide. Alkaline slant, acid butt, hydrogen sulfide, and then gas bubbles. Last reaction K by K. Alkaline slant and acid butt. No reaction. So everything negative in this case. So these are the different original media. First one is control. No reaction takes place in this medium. The medium is exactly like an inoculated medium. No reaction. Next one is E. coli. The E. coli appear like this. A by A. Alkaline slant, acid butt. Next is Salmonella typhi. It may appear K by A. That is alkaline slant, acid butt plus H2S. Black color. Next, fourth one is Proteus vulgaris, which means alkaline slant and also acid butt. Acid, the yellow color is not seen. Why? Because of the hydrogen sulfide production. Here it is yellow color, but it is not seen because of the hydrogen sulfide production. This hydrogen sulfide production takes place in the acidic environment. Okay, so it is denoted as K by A with H2S. H2S is heavily produced in Proteus vulgaris. Okay, then next is A by A with H2S. That is Citrobacter fronti. Here acid slant, acid butt with hydrogen sulfide. Here also another reaction similar to fourth one. That is Proteus vulgaris, K by A. Sixth one, Salmonella typhimurium, alkaline slant, acid butt and H2S production. Next is Shigella flexneri, K by A. K alkaline slant, A acid butt. No H2S production, no gas bubble, etc. So these are the reactions. So here is the interpretation given. Red or K by A, that is glucose fermentation only. But is alkaline, acid. Glucose fermentation only, peptone is catabolized. A by A, glucose, lactose and everything is fermented. K by K, no reaction. A by AG, that is glucose and lactose fermented or sucrose fermented and with gas production. That is A by AG. K by AG means glucose fermentation only. Alkaline slant is alkaline. So, but gas is produced. Here also glucose is fermented. If the glucose is only fermented, the butt will remain only acid. The slant is remain alkaline. In that case, glucose is only fermented. K by A. Gas produced, H2S production. A by A means glucose, sucrose, everything fermented with gas, H2S. Okay, so these are the interpretation of TSA. Here also. Next is Christensen's urea medium. This urea agar, it is used to detect urease production. And this urea agar described by Christensen detected urease activity by all rapidly urease positive Proteus organisms and also by the other members of the Enterobacteriaceae that exhibit delayed urease reaction. So mainly this urease reaction is positive in the Proteus species, that is coming under Enterobacteriaceae family. This was accomplished by adding glucose to the medium, decreasing the peptone concentration, decreasing the buffering system, as less buffered medium detects small amount of alkali. That is why the delayed urease reaction. Peptone is the source of essential nutrients in the urease medium. Dextrose is the energy source. Sodium chloride maintains osmotic equilibrium, and the urea is hydrolyzed to liberate ammonia. Phenol red indicator is used to detect alkalinity generated by visible color change, and the color changes from orange to pink in color. Prolonged incubation may cause alkaline reaction in the medium. The medium without urea serve as negative control. So rule out the false positive reaction. Also, urease tests relay the alkalinity formation. They are not specific for determining absolute rate of urease activity. Utilization of proteins may raise the pH of the alkalinity due to the protein hydrolysis and excess of amino acid liberation results in false positive results. So, whenever the proteins are utilized, the pH can be raised, which may result in alkalinity. This is the reaction. Negative test will be appear as this color, positive pink in color. Next is bordered JNCO medium. It is a type of agar plate. It is used to isolate Bordetella, and it is mainly used to isolate the organism Bordetella pertussis, causative agent of pertussis. It contains blood potato extract and glycerol with an antibiotic cephalexin or penicillin. The potato extract, which provide nitrogen and vitamins, and potato starch absorbed fatty acids present in the nasal secretions or collection swab cotton that inhibited growth. Glycerol was the carbon source. And the medical microbiology 4th edition states that Reagan-Low medium contained charcoal, blood and antibiotic, and replaced bordered JNCO medium as the medium of choice for the routine Bordetella pertussis incubation. And this organism is very difficult to culture. So they invented the first version to isolate Coccobacillus named bordered JNCO in 1906. They believed was associated with the whooping cough, and it was not yet established Coccobacilli was only the cause. This is the bordered JNCO agar, and the organism Bordetella pertussis. Last one is thiosulfate citrate bile sucrose medium. It is mainly used to isolate Vibrio cholerae. The Vibrio cholerae appear as a yellow color in this agar, and the other species Vibrio parahemolyticus appear as dark green color. As a result of the fermentation, the color changes to yellow. This is the TCBS agar, type of a selective agar used in the microbiology laboratory to isolate Vibrio, and it is highly selective medium for isolating Vibrio cholerae from Vibrio parahemolyticus. And it contains high concentration of sodium thiosulfate and sodium citrate, which inhibit the growth of Enterobacteriaceae family and also inhibit the gram-positive bacteria. And it can be achieved by the incorporation of oxgal. It is a naturally occurring substance containing a mixture of bile salts and sodium chloride. It is a pure bile salt. The sodium thiosulfate serves as a sulfur source, and its presence in combination with ferric citrate, it helps in the easy detection of hydrogen sulfide production in Vibrio cholerae. Saccharose, that is sucrose, is included as a fermentable carbohydrate for the metabolism by Vibrio species. And the alkaline pH of the medium, it enhances the recovery of Vibrio cholerae because Vibrio cholerae is more alkaline loving, and it inhibits the growth of others. Here, thymol blue and bromothymol blue are included as indicators. So, the expected results, Vibrio cholerae can appear large yellow colony, Vibrio parahemolyticus blue to green centers, and Vibrio alginolyticus, they are also large yellow mucoidal colonies, and Vibrio harvey fishneri, grayish green to bluish color. So, these are about Vibrio cholerae and also culture media.